Similar to the link reaction, the Krebs cycle takes place in the mitochondrial matrix. It is a series of enzyme-catalyzed reactions producing two molecules of carbon dioxide while conserving energy by reducing the coenzyme NAD, which we've talked about before, and a second coenzyme called FAD, which stands for flavine adenine dinucleotide. These reduced coenzymes then carry the hydrogen atoms to the electron transport chain in the cristae, where they will be used in the production of many more ATP molecules. The Krebs cycle unfolds through the following steps. Step 1. The acetyl group released from acetyl coenzyme A combines with a 4-carbon compound, oxaloacetate, to form a 6-carbon compound called citrate. Step 2. Citrate is decarboxylated and dehydrogenated, producing a 5-carbon compound. 1 molecule of carbon dioxide and 1 molecule of reduced NAD is produced. Step 3. The 5-carbon compound undergoes further decarboxylation and dehydrogenation, leading to the production of a 4-carbon compound. 1 molecule of carbon dioxide and 1 molecule of reduced NAD is produced. Step 4. This 4-carbon compound combines temporarily and is then released from coenzyme A. At this stage, substrate level phosphorylation takes place, producing 1 molecule of ATP. Step 5. The 4-carbon compound is dehydrogenated, producing a different 4-carbon compound and a molecule of reduced FAD. Step 6. Rearrangement of the atoms in the 4-carbon molecule, catalyzed by an isomerase enzyme, is followed by further dehydrogenation, regenerating a molecule of oxaloacetate so the cycle can continue. So to summarize, for every turn of the Krebs cycle, this results in the production of 1 molecule of reduced FAD, 3 molecules of reduced NAD, and 2 molecules of carbon dioxide. However, because every glucose molecule yields 2 acetyl groups, therefore all of these products would double. As a result, 2 molecules of reduced FAD are produced, 6 molecules of reduced NAD are produced, and 4 molecules of carbon dioxide are produced, as well as 2 ATP molecules. In the production of ATP, I refer to this as substrate level phosphorylation. This is when the phosphate group is directly transferred from one molecule to another. The coenzymes produced in the Krebs cycle play a crucial role in the subsequent stages of cellular respiration, particularly in the electron transport chain, where they contribute to the synthesis of ATP.
Hi, my name is Mr. Science, aka Salim. If you're new to the channel, please remember to like and subscribe, and for more teaching resources, you can visit my website at www.mrscience.co.uk.